Everyone, and welcome to the May FightMND Scientific Seminar. My name is Sarah Bennett, and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of the month, and last month in April, we heard from Associate Professor Cheyenne No from the University of Queensland. She spoke about her recently published Metflex-1 clinical trial and the journey of preclinical discovery that had led to that point. If you would like to know more about this and you missed Shu's talk, it is available on our website to view at your convenience. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note, however, that the chat function is only enabled if you're using an app-based version of Teams and unfortunately doesn't work in the web-based version. So please ensure you introduce yourself and where you're from before asking your questions so we can all get to know each other. We are recording today's presentation, so if you would know anyone who's not able to attend, they will be able to watch it back in a few days' time. So now to our presentation for today. Dr Jeremy Lum is a Bill Gole MND Postdoctoral Fellow in the School of Medical, Health and Indigenous Science at the University of Wollongong. He received his PhD in cellular neuroscience and neuropharmacology from the University of Wollongong in 2019, and he currently leads an arm of the late Professor Justin Yerbury's research team, where he is utilising cell and mouse models of MND to advance the investigation and assessment of therapeutic avenues. Jeremy joins us today to discuss some recent breakthroughs in the Yerbury lab. Thank you so much, Jeremy, for joining us today, and over to you.
Thanks so much, Sarah, and FightMND for inviting me to talk today. And I'd also like to just acknowledge the traditional land and knowledge holders of the Dharawal country where the University of Wollongong is located. So today I'll be talking about some work that we've been doing in Professor Justin Yerbury's lab that has been funded primarily through FightMND. And I joined Justin's lab about 4 to 5 years ago, particularly working on a therapeutic arm of his team, focusing on targeting SOD1-MND. So typically I'd start my talks outlining ALS or MND, but this audience is far too familiar with this disease. So I'm going to start talking about cancer a little bit, not an area that I'm a particular expert, but the field of cancer has come a long way in the approach that they treat people, with survival rates continually on the rise. So typically patients won't receive just one type of therapy. They'll receive a combination of therapies, including surgery, a cocktail of chemotherapy, drugs, radiation, immunotherapy, and targeted therapy. And this kind of underlies the therapeutic efficacy in cancer. Now, not only do these drugs act independently, but they also, when used together, act in an additive or synergistic manner to reduce the tumour size. Now, we believe that using this kind of approach underlies that therapeutic efficacy and is something that we've taken in our approach to treat SOD1-MND. So here's a slightly outdated figure of a list of genes that have been associated with MND or ALS. And you can see that that number's continually rising. However, our interest in the Yerbury lab is particularly on this SOD1 gene, which affects about 2% of sporadic ALS patients and about 20% of familial ALS patients, including Justin, who's pictured here with maybe arguably two of his favourite lab members, Luke and Nat. But if we take a closer look at that SOD1 gene, every little bubble here represents a gene, a variant in the SOD1 gene that has been associated with MND. And there's roughly been about 230 variants that have been associated, which is quite significant considering that the SOD1 gene is not that large. So like most mRNA, it is the template for producing the SOD1 protein. So here we have nascent SOD1 that comes off the ribosome. Now, under physiological conditions, this will push down a SOD1 folding pathway where it undergoes numerous post-translational modifications until it reaches its final folded state as a homodimeric enzyme that's responsible for neutralizing free radicals in our cells. Now, a unifying feature of mutations within SOD1 out of all those 230 is that what it does is it actually pushes SOD1 down a misfolding pathway at various points. And these misfolded proteins or misfolded SOD1 is what we believe is quite toxic to the motor neurons. And you can see here when we look at post-mortem tissue from MND cases or SOD1 MND cases, you can see using antibodies that specifically bind to misfolded SOD1 that we see a high presence of the misfolded SOD1 in motor neurons. Furthermore, this is another case here from a SOD1 patient or post-mortem tissue, and we can see this large SOD1 inclusion, which represents the final kind of stages of that SOD1 misfolding process. And these are a hallmark of SOD1 post-mortem tissue. Now, I don't have time to go through all of Justin's findings over the last decade or so, but Justin was quite interested in understanding how misfolded SOD1 species affect cellular processes. And what he found was that it affected numerous cellular processes in the body, all linked to MND, including mitochondrial dysfunction, ER stress, excitability, protein quality control. Now, Justin used to say that MND was almost like death by 1,000 cuts, that this misfolded SOD1 species would affect so many different cellular pathways shown here, and that each cellular pathway almost affected the other ones as well. But ultimately what happens is all these disrupted cellular processes lead to the degeneration of the motor neuron. But at its core, we strongly believe that it's this the mutations within the SOD1 gene that push it down this misfolded SOD1 pathway and cause the production of misfolded and aggregated SOD1 that is the cause and leads to the degeneration of these motor neurons. So recently, SOD1 MND has made quite a bit of headway in the way that it can be treated, particularly through the generation of tofersen, which is a SOD1 ASO or antisense oligonucleotide. Now, tofersen works by binding to SOD1 mRNA and facilitating its degradation so that we get less of the protein. Now, this is a figure from the Valor trial, which was initially only meant to go for 28 weeks. And we can see here in blue, these are patients that received placebo, and then in red, patients that received tofersen. And you can see that after 28 weeks, the results don't show as much clinical effectiveness as was originally thought, particularly when we look at survival. However, there was an open label extension trial, and it was quite clear that tofersen did have a clinical effect, most notably improving survival after 52 weeks of treatment. So tofersen, for me at least, represents probably the greatest achievement so far in treating SOD1 MND. And we might kind of think the job is kind of done there. But we believe that there are some challenges that tofersen faces, and there's opportunity to move forward with this. So one of those challenges with tofersen is that it actually requires quite regular intrathecal injection of the ASO into the spinal cord. So if this can obviously lead to sometimes adverse events and is quite invasive. So if we were able to deliver this peripherally, that would be a significant advantage. Furthermore, from the Valor trial, when we look at the cerebrospinal fluid, the SOD1 levels in the CSF, what we're able to see here is that even those patients that were on tofersen throughout the whole trial, it only reduces SOD1 protein expression in the CSF by about 30%. So if we go back to this SOD1 folding pathway, what that means is that 70% of the protein of the mRNA is able to be translated into potentially these misfolded protein species. So we obviously want to improve on that. So one thing that we've been doing is in collaboration, and it's really being led by Cara Vine-Perra here at the University of Wollongong. Her and Justin came up with this idea a while ago, and they've published this paper, I think, over 5 years ago, where they were able to encapsulate these SOD1 ASO particles within calcium phosphate nanoparticles. And what they show is that by encapsulating the ASO within these nanoparticles, you're able to increase the circulation of the ASO and also the uptake within neuronal cells to downregulate that SOD1 protein expression. But if we're going to deliver tofersen peripherally, what we also need to overcome is the blood-brain barrier, which is tightly regulated by these tight junctions to stop things just passively going across the into the brain. So to overcome this, what we've been using is focused ultrasound technology. So for those of you that are unfamiliar, focused ultrasound is actually being used clinically for tremor and Parkinson's disease. And what it involves in this mouse model here is delivering these little gas-filled microbubbles into the circulation. And the microbubbles will travel through the mouse all the way into the microvasculature that surrounds the brain. What we can then do is apply stereotactically guided ultrasound onto the brain, and that activates these microbubbles so that they start to contract and expand. And they will mechanically open this blood-brain barrier. And hopefully what will happen is this will allow us to potentially deliver these SOD1 encapsulated ASO particles into the brain and allow better delivery of that ASO. So to test this, two postdocs up in Kara's lab performed an acute study where we just wanted to see whether we could increase ASO delivery to the brain using this focused ultrasound technology. So here on the top row, we have a mouse that just received the naked ASO. And this is some MRI images with gadolinium contrast. And you can see that there's quite minimal disruption of the blood-brain barrier or entry into the brain. And we also, I should mention that the ASO is labeled with a sci-fi tag. And there's quite minimal ex vivo imaging of the brain, shows quite minimal sci-fi signal. However, when we use focused ultrasound with the ASO, you can see from the MRI images, we do get disruption of that blood-brain barrier. And when we look at the sci-fi signal, we can see that there's a relatively higher amount of this sci-fi signal in the brains that receive focused ultrasound, suggesting that there's increased penetration of that ASO into the CNS. So we then took this into a mouse, the SOD1 G93A mouse model, and gave them a 9-week treatment of this focused ultrasound and ASO, which we deliver peripherally. And we gave them either focused ultrasound alone or the ASO alone, or we combined the two together. And here we have some brains of those mice where we've stained for SOD1 shown in red and DAPI in blue. And when we quantify the amount of SOD1 intensity in the brain or in the cortex of these mice, we can see that we can drastically reduce SOD1 expression when we combine the ASO with focused ultrasound technology. And interestingly enough, when we perform motor neuron counts in the spinal cord of these mice, and we observed that our focused ultrasound ASO group had significantly more motor neurons than those that just received either focused ultrasound or the ASO alone. So this provides us with some encouraging results, and we're currently following this up at the moment to look and see whether it can have any modifications on disease progression and survival rates. So back to our SOD1 pathogenic pathway, we've kind of started at the top here. And like I said before, tofersen clinically only reduces SOD1 levels in the CNS by about 30%. So it still allows a lot of this toxic SOD1 to potentially be produced. So what Justin kind of thought of was maybe we can push some of this mutant SOD1 to correctly fold. So to do that, what we started to do was look at the SOD1 folding pathway, which is shown here. So this is the correct SOD1 folding pathway. Now, the SOD1 protein requires, when it comes off the ribosome, to undergo some spontaneous folding before zinc can be delivered to it. Once zinc is bound, it moves further through the pathway and copper is able to be delivered to the SOD1 protein via the copper chaperone protein. Then following this, we have an almost mature monomeric version of SOD1. However, it requires the oxidation of these two free cysteine residues to form an intermolecular disulfide bond. So once that occurs, we can then have a mature monomer, and these two mature monomers can join together to form the fully functional and stable homodimeric SOD1 protein. Now, what we do know after looking at SOD is that as SOD1 moves through this pathway, it actually becomes increasingly more stable. And this homodimeric fully folded SOD1 is actually one of the most stable proteins in the body. So what we also do know is that when there are mutations within the SOD1 gene that carries through to the protein, it disrupts this folding pathway and leads it into off-folding pathways, and that's where we get the accumulation of that toxic misfolded SOD1. So what we were trying to do was look for molecules that would push SOD1 through the folding pathway. And one molecule that you might be familiar with due to its recent testing in an ALS clinical trial here in Australia is copper ATSM. Now, Justin believed that copper ATSM may be able to help deliver copper to the SOD1 protein and push it down this normal folding pathway. So to test this, Natalie Farrell in our lab took NSC34 cells, which are a motor neuron-like cell. She transfected them with a bunch of different SOD1 mutants, and that's tagged with an EGFP. We then can treat the cells with copper ATSM and look at the microscopy, look at SOD1 inclusion formation via microscopy. Now, I should point out there are two types of SOD1 mutations. The first being metal binding region mutants, so they disrupt copper and zinc binding. And then there's wild type like mutations which are outside those regions. And what we observed was that these wild type like mutations are represented in red here. What we observed is that when we add copper ATSM to these cells, we get a much lower number of inclusions compared to those cells that did not receive copper ATSM at all. And that was the same trend across all the wild type like mutants. However, when we transfected cells with a metal binding region mutant and treated them with copper ATSM, we don't see any difference in the number of inclusions formed. Furthermore, when we look at the viability of these cells, we also note that the wild type like cells had increased survival when they were treated with copper ATSM. However, when we looked at metal binding region mutants, there was no difference in survival. Now, this kind of gave us something to think about in the fact that potentially one of the mechanisms copper ATSM may be working is via delivering copper to the SOD1 protein and pushing it along that pathway. But also, it might be useful to think about this from a clinical perspective. Potentially, patients harboring a SOD1 metal binding region mutation, they may not respond to copper ATSM. So, copper ATSM had been through, at the time that we started to look at it, had shown in various SOD1 mouse models that it provided neuroprotection and increased lifespan. So we decided to test this in our own hands, and we teamed up with Peter Crouch to do this. So what we did was we had our SOD1 G93A mouse model, and we delivered a relatively high dose of copper ATSM, so 100 milligrams per kilogram per day, to these mice. And when we look at the body weight change, you can see that the vehicle mice are slowly putting on weight at the initial stages of disease. However, when we treated our mice with copper ATSM, they actually started to lose weight, which was quite surprising. And what we noticed was that the mice that were receiving copper, this relatively high dose of copper ATSM, they actually started to show quite toxic-like symptoms. grimacing quite hard, bit of piloerection here, or a lot of piloerection, indicative of an adverse event or toxicity. So what we did was we decided to drop our dose of these mice. We gave them a 2-week washout period after seeing this, and we decided that we would start to drop the dose to 60 milligrams per kilogram per day. And similar to the studies that Peter Crouch and others had shown, we were able to show that copper ATSM delays disease progression in these mice. So we have neurological score on the side, with 4 being full paralysis, 0 being no symptoms at all. In the blue, we have the vehicle, and the copper ATSM mice, we were able to see a slowing of disease progression. And furthermore, when we look at the survival of these mice, we also noted that their copper ATSM increased their survival. Furthermore, we ran Western blots on the lumbar spinal cord looking for SOD1, and what we were able to show was that copper ATSM increases the solubility of the SOD1 protein in the spinal cord, suggesting that it's increasing its stability there. So there was kind of 2 things that we got out of this. One, that high levels of copper ATSM can be quite toxic, which had been reported in the phase 1 clinical trials, but also that SOD1 does, when used at the right dose, can increase the solubility and the stability of that SOD1 protein in these mice. So going back to our SOD1 folding pathway, Luke and Victoria Shepherd in our lab became quite interested in potentially other molecules that might be able to shift SOD1 through this maturation pathway. And one compound that we became quite interested in was Epsilon, which is an organoselenium-based drug and has been used in clinical trials before for other diseases. And what Epsilon was thought to do was help with the oxidation and the formation of this intramolecular disulfide bond. So what they did was they transduced NSE34 cells with wild type like SOD and treated them with either a vehicle or Epsilon. And you can see here there are 2 bands. Now this top band here represents the reduced form of SOD1, immature form of SOD1, whilst this band here represents this intermolecular disulfide bond and more mature form of SOD1 shown here. And what they were able to show was that when you treat the cells with Epsilon, you actually shift the amount of, or the proportion of SOD1 that's getting pushed through this pathway. Furthermore, they repeated that experiment in SOD1-A4V transfected cells and showed the same result. So they also looked at inclusion formation. So here we have SOD1-A4V, which is a wild type black mutant, and SOD1-G85R, a metal binding region mutant. And they were able to show that Epsilon dose dependently increased cell viability when treated with Epsilon. Now, at the same time as we were doing this work, Neil Cashman over in Canada, a longtime collaborator of the lab, had become quite interested in this tryptophan 32 residue shown here, which becomes solvent exposed when SOD1 goes through its misfolding pathway. So to look at this further, what Neil did was purified wild type SOD1 with the tryptophan 32 residue, and you can see here in blue, SOD1 via a THT assay fibrillates quite quickly and aggregates, it increases the aggregation propensity. However, if you substitute that tryptophan 32 residue with a serine in this case, you actually lose the aggregation propensity of SOD1. He also repeated these experiments in SOD1-G93A and V148G and showed that we lose that effect when that tryptophan 32 residue is substituted with a serine. So Justin became quite interested in, well, can we block that tryptophan 32 residue to potentially block the aggregation of misfolded SOD1? So one molecule that Neil found was telbivadine, a pyrimidine-based retroviral inhibitor. And he did some computational modeling showing that it could potentially bind to this tryptophan 32 residue and block its aggregation. So we took it into a zebrafish model of harboring a SOD1 G127X mutation. And when you treat these zebrafish with telbivadine, you can see that there's a dose-dependent increase in low-to-mocha function, suggesting that telbivadine can actually protect against SOD1 toxicity in vivo. So collectively, over the aforementioned studies, what we started to see was that potentially we had multiple tools to target this SOD1 misfolding and aggregation that could potentially help with copper ATSM, which was in the clinic at the time. So we decided to use this as a potential polytherapy approach, where we use all these three drugs in combination to help aid SOD1 folding and block aggregation. So to test this, we went back to our cell model. So we used NSC34 cells, transfect them with SOD1 G93A, and then we run a 3D checkerboard assay where we can use increasing concentrations of one drug along the plate, then down the plate, increasing concentrations of the second drug, and then across multiple plates, increasing concentrations of that third drug. So this work was done by Natalie Farrell in our lab. And here we have the fold change in cell survival. And you can see running up the y-axis, copper ATSM dose-dependently increases cell survival. Furthermore, along the x-axis here, you can see that Epsilon also dose-dependently increased cell survival, which was in line with our previous results. But we also noticed that we can use these drugs at particularly copper ATSM, at much lower concentrations to produce better therapeutic efficacy than any one drug alone. Furthermore, when we look at telbivadine, it also showed some modest increases in cell survival. And what we ended up doing is picking a concentration ratio here that we decided to carry forward. I won't go into too much detail about how we got to this ratio for time's sake, but we decided to use this ratio here. So here we have some cells that have been treated with our polytherapy or copper ATSM alone, and where we've measured the number of inclusions within these cells. And you can see that copper ATSM dose-dependently reduces inclusion formation. However, when we use our polytherapy, and I should note that the concentration of copper ATSM in this polytherapy is half of what is shown at the highest dose of copper ATSM, we actually get the same level of reduced inclusion formation, suggesting that we can actually use copper ATSM at lower doses in combination with Epsilon and telbivadine to produce the same therapeutic effect as higher concentrations of copper ATSM. Furthermore, just to show that we are increasing the proportion of SOD1 maturation, we performed that disulfide bond assay, and you can see here that our polytherapy outperformed even the highest concentration of copper ATSM tested in producing that intermolecular disulfide bonded SOD1. So based on this, we decided to take it back into our animal model. So we used a SOD1 G93A mouse model, and we treated the mice either with vehicle. We compared it against copper ATSM treatment alone, as we thought this was kind of the gold standard in targeting SOD1 at the time. And then we used our polytherapy, where the dose of copper ATSM is the same as that of those mice treated alone. And similar to previous work, we showed that copper ATSM slows disease progression compared to the vehicle group. However, when we treat with our polytherapy, we show that we're able to slow disease progression even further. We also showed that our polytherapy approach was able to delay disease onset compared to both copper ATSM and vehicle-treated mice. Looking at the survival of these mice, we again repeatedly shown that copper ATSM increases the survival of these mice. Furthermore, when we look at our polytherapy, it also shows a modest improvement in survival compared to those mice treated with just copper ATSM alone. So to follow this up, we looked at the spinal cords of these mice and measured the relative solubility of SOD1. And we can see here that the polytherapy was able to increase the levels of SOD1 in the spinal cord of these mice compared to the vehicle group. And also when we look at the proportion of the disulfide bonded SOD1, we see that our polytherapy also improves on the vehicle treated controls, which only backs up that our polytherapy is able to improve SOD1 maturation. Quite interestingly, we then performed a dot plot on these mice, on the lysate, spinal cord lysates of these mice, specifically looking at probing with an antibody C4F6 that binds to misfolded SOD1. And what you can see is that whilst copper ATSM does reduce the levels of this misfolded SOD1 to some extent, the polytherapy was able to drastically reduce the accumulation of that toxic SOD1 species. And furthermore, when we perform motor neuron counts on these mice, we see that whilst copper ATSM did provide protection in motor neuron loss, the our CET polytherapy was slightly better. So to validate these findings, we actually repeated the study again, or a very similar study, but we dropped the copper ATSM dose to 25 milligrams. So from 50 milligrams, we dropped it down to 25 milligrams. And we also wanted to ensure that our polytherapy was compatible with riluzole. And what we observed, just going quickly through this, is that riluzole had very little effect, slightly improved actually, the CET polytherapy, but it definitely didn't make things worse. And then when we look at the survival as well, we see that CET and our CET with riluzole was able to increase the survival of these mice compared to vehicle. So it would suggest that our polytherapy is compatible with riluzole, and riluzole may slightly improve its efficacy. Now, here I have a graph of the percent survival increase of all the studies that we did with copper ATSM. And I kind of want to bring your attention particularly here to these two, where this is from our initial study where we used 50 milligrams per kilogram of copper ATSM, and we see about a 6% increase compared to the vehicle control. However, when we use copper ATSM at 25 milligrams per kilogram in combination with epsilon and telbivadine, we see that we are able to get a similar level of therapeutic efficacy, suggesting that we can use lower doses of copper ATSM with epsilon and telbivadine to produce similar therapeutic outcomes to higher doses of copper ATSM. And that's particularly important when we think about the adverse events that we saw with those relatively high doses of copper ATSM before. So this is the last part of our study. What we'd kind of shown is that we can, or the field has shown, is that we can silence SOD1 expression through with the use of ASOs, and that we had shown that we can help stabilize SOD1 using a polytherapy approach of small molecules. But Justin was quite interested in misfolded SOD1, and particularly he had shown previously that misfolded SOD1 can be ubiquitinated and degraded primarily via the proteasome. So we became focused on how can we help aid this pathway. So we did this through the use of proteolysis targeting chimeras, or PROTACs, which some of you may be familiar with. So what that is we've got a ligand which binds to a chemical ligand which binds to the protein of interest, and you can stick an E3 ubiquitin ligase on the other end of it with a linkage joining the two. And the aim of the game is that this protein ligand can identify a protein of interest, and the E3 ligase can facilitate the degradation of this. So here we have a picture of the ubiquitin proteasome system, and it illustrates here that what the PROTACs can do is bring the E2 ligase in closer proximity to the protein of interest and help add a ubiquitin signal onto that protein to facilitate degradation. So whilst chemical PROTACs are quite good when there's a binding pocket for the protein, misfolded SOD1, as far as we know, does not have a binding pocket. So we kind of had to tweak this approach. So to do that, we teamed up with Professor Neil Cashman at the University of British Columbia, and now has a spin-off company, ProMUS Neurosciences. And he was working on humanized antibodies for misfolded SOD1. And I've got one of his papers here where he's generated a humanized antibody that's able to detect misfolded SOD1, specifically misfolded SOD1, in motor neurons of a mouse model. So what I should point out here actually, that Kristen Chisholm here, who was a PhD student at the time and is now, we're lucky enough to keep her as a postdoc, has been working on this. So what she was able to do was take the SCFV portion of this misfolded SOD1 antibody, and then she attached an E3 ligase on the end. I should point out as well that we do have a hiss and a flag tag on this what we're going to call a bioprotac. So it works the same as a protac, just for downstream experiments. And the idea is that the SCFV portion would specifically recognize misfolded SOD1, and with the attachment of that E3 ubiquitin ligase, it would facilitate the degradation of the misfolded SOD1 to the proteasome. And Kristen just didn't do this with one of Neil's antibodies. She actually did it with 7 different antibodies that Neil kindly gifted us. And then as a control, we actually had an SCFV, which was targeting beta-galactase as our negative control. So here we have some cells. What we did was in a cell line, we co-transfected in this case, SOD1-A4V mutant with an EGFP tag, and we also co-transfect in our bioprotac. And then what Kristen did was perform what's called a saponin assay. So saponin's a detergent-like chemical, and what it does is punch little holes into the membrane of these cells. And what that does is allow all soluble material to exit the cell. And then when you image the cells, what you were imaging is the insoluble SOD1 that was unable to exit those little pores. And so you can see here images of those cells. So what you're seeing here is the EGFP signal representing a insoluble or aggregated SOD1. And you can see that almost all of our bioprotacs were able to reduce the number of aggregated SOD1 in these cells. Particularly, we had 1, 2, 3, 4 bioprotacs that were able to do this by more than 50%. Now, Kristen ran a whole bunch of assays to start to screen these bioprotacs. However, for time's sake, the quick and dirty answer is that we settled on bioprotac 2. So as I mentioned before, SOD1 has over 230 mutations, and we wanted to test whether the bioprotac would work on all of these. So she performed the same assay in cells transfected with different, I think 9 different SOD1 mutations. And what we see is that we see relatively good efficacy in reducing the amount. This is shown here, the reduction in insoluble SOD1. We see that our bioprotac 2 was able to do this across a range of SOD1 mutations. Furthermore, to show that the bioprotac was actually binding to mutant misfolded SOD1, we ran some immunoprecipitation experiments, and you can see here that this is our control bioprotac against beta-galactase. When co-transfected with SOD1-A4V, we don't get any pull down of any SOD1 in the cell. However, when we immunoprecipitated our bioprotac 2 with A4V, we pulled down a relatively high level of SOD1 shown here in the immunoblot. Now, we also tested it against wild type SOD, and we see that we do get some minimal SOD1 pull down. But that's not unexpected, considering that we have shown previously that overexpression of SOD1 wild type can lead to misfolding as the cell's unable to keep up with its folding process. So we then wanted to ask the question about our bioprotac is, does it need that E3 ubiquitin ligase on the end? Because other groups had shown that you can just inject either an antibody or an scFv in SOD1 mice directly into the brain and downregulate misfolded SOD1 levels. So to do this, Kristen created another construct. Now, I should point out, CHIP is our E3 ubiquitin ligase that we used. She generated the bioprotac with and without the E3 ligase. So firstly, if we look at the, she then co-transfected it with SOD1 G93A as well. And what you can see is that the bioprotac by itself, so lacking that E3 ubiquitin ligase, whilst it does reduce some of the relative SOD1 levels, the bioprotac with the ubiquitin ligase downregulates those SOD1 levels to a much greater extent, suggesting that that E3 ubiquitin ligase is integral to the action of our bioprotac. We also hypothesized that that our bioprotac was facilitating degradation through the proteasome. So to test this, what we did was we tested these two bioprotacs with and without the E3 ubiquitin ligase in the presence of the proteasomal inhibitor MG132. And you can see the action of the bioprotac without the E3 ligase is inhibited to some extent, whilst the is not inhibited, whilst the action of the full-length bioprotac is inhibited by MG132. Furthermore, we wanted to look at the other degradation system, the autophagy system, and we kind of see almost the inverse results, where the bioprotac without the E3 ubiquitin ligase is somewhat inhibited by the autophagy inhibitor baflamycin. So this kind of suggested to us that the bioprotac itself was degrading through the proteasomal system, whilst the bioprotac or the scFv portion primarily works through the autophagy system. And then at this point, what we decided to do was name our protac, and Justin came up with this nice name called misfoldable, because what our bioprotac does is target the degradation of misfolded SOD1 via the ubiquitin ligase pathway. So we wanted to take this into mice. So we teamed up with Lars Ittner and Fabian Delarue at Macquarie Uni, and they were kind enough to generate a CRISPR mouse model where the misfoldable expression was under the human synapsin 1 promoter. And we generated this misfoldable mouse line. And unfortunately, when we crossed these misfoldable mice with SOD1 mice, hoping to see that the misfoldable SOD1 double transgenic mice would have some sort of neuroprotection, unfortunately, when we look at the survival rates in both female and male mice, we didn't see the neuroprotection that or the disease modifying effects that we were hoping for. However, something quite interesting when we analysed the brains and the spinal cords of these mice is that I'll point you to this this section of the graph here. The misfoldable double transgenic mice shown in blue actually had lower levels of insoluble SOD1 compared to the SOD1 mice in general. However, we didn't see that same effect in the spinal cord. We also were able to observe that our misfoldable mice actually had more innervated NMJs compared to the SOD1 mice. Furthermore, we were able to show that these SOD1 misfoldable mice had an increased amount of motor neurons compared to the SOD1 wild type mice, suggesting that there is some form of neuroprotection. Now, to follow this up further, we actually went and wondered, well, why are we having an effect in the brain and somewhat in the spinal cord? And what we observed is that the misfoldable, this here is a wild type mouse. This here is our misfoldable, where we've done immunohistochemistry on the flag tag. And you can see that we get quite good expression of that misfoldable in the cortex. And immunoblots of the spinal cord and brain of the misfoldable mice showed that we get an extremely high amount of the misfoldable in the brain, but very low expression in the spinal cord lysates of this mice, suggesting that synapsin 1 is probably not a good promoter for spinal cord expression. So what we've done now is we've teamed up with Yazi Key's lab and Annika Van Humble at Macquarie Uni. And what we're actually doing at the moment is we've modified the promoter and we've actually put it into an AAV system and currently running a series of experiments to see whether AAV delivery of the misfoldable gene can protect in the SOD1 mouse model. And this study is currently ongoing and something that I hope that we can present in future meetings. So to finish, SOD1 ASO delivery can be improved with the use of focused ultrasound, and that small molecules can improve SOD1 folding and reduce misfolded and aggregated SOD1 load. Furthermore, bioprotacs offer a potential avenue to degrade misfolded SOD1. But ultimately, I hope what you get from this talk is that utilizing such therapies in a multifaceted approach or a polytherapy approach may be more robust, may provide more robust benefits for the complex pathology of SOD1 and the entire MND landscape. And with that, I'd just like to thank you for listening. A big thanks to Luke, Kristen, Nat, Michaela, and Rach, who performed a lot of this work. Obviously, a big thank you to Justin, who's pretty much given us the ideas to move forward with. All the collaborators shown here. And I'd also like to pay a special mention to what we call our Brains Trust here at UAW. In particular, Heath Eckroyd, Cara Perro, Lizanne Uy, Rachel Yerbury, and Darren Saunders, who have really made sure that the Yerbury group has been able to stick together and continue to grow. And of course, lastly, I'd like to thank FightMND, MNDRA, and the NHMRC for funding a lot of this work.
Thank you so much, Jeremy. It was a really fantastic talk that really covered a lot of different facets of the group's work. So does anyone have any questions? We're open the floor to questions right now. There should be a small raise hand button at the top of your screen that you can press, and we'll be able to enable your microphone and camera. Or if you'd like, you can also type your question into the chat and I can read it out on your behalf. So maybe while we're waiting for any questions, Jeremy, I'd just like to start off by going right back to the start where we talked about the focused ultrasound delivery of the ASOs. And I was just curious as to whether there have been other examples of focused ultrasound to deliver therapies in other indications in the clinic.
In the clinic?
Yeah.
Yeah, so focused ultrasound is being used for tremor and for Parkinson's disease, but it's mostly just the application of the focused ultrasound, so not necessarily delivering anything else. I think Sydney has actually opened up 2 focused ultrasound clinics in the last couple of years as well, so it is clinically translatable.
Amazing. Just another question: have you got experiments planned to sort of bring together everything that you've spoken about, trialing all of these different facets together?
Yeah, it's definitely something that we want to start to do. We still got a little bit of optimizing to do. We can definitely do it in cells, but taking it forward into an animal model might be a little bit harder. But yeah, it's definitely something that the group is definitely keen to pursue.
Great. I just have a question in the chat. It's from Gethin Thomas. It says, I might have missed it, but did the copper ATSM riluzole combination treatment start before or after symptom development in the mice? Yeah,
it started at about 50 days old, so it is a pre-symptomatic proof of concept treatment.
Great. I have another question in the chat from Sunny Ann. She says, firstly, great talk. Secondly, she has a couple of questions. I'll just start with the first one. In addition to motor neurons, have you looked into the phenotypes of non-neuronal cells, such as microglia and astrocytes, after the polytherapy approach?
No, but it's definitely something that we would be interested in doing. Copper ATSM has shown an effect, particularly in astrocytes, I believe. So yeah, it definitely is something that we would be interested in doing, but no, we haven't looked at non-neuronal cells.
Thanks. And just a second question from Sunny Ann. In vivo copper ATSM, the copper ATSM mice reduce the total weight. Have you looked into the fat versus lean muscle mass from them? And have you looked into the hypothalamus between the groups?
No, we haven't looked peripherally. We could definitely look in the hypothalamus. We do still have that tissue that we could dissect out, but no, we didn't look at the fat to body fat and muscle mass ratio.
Thanks. Any other questions? Yep, I will. Gethin, you should be able to turn your camera and mic on if you'd like.
I won't bother with the camera, but the mic. Hey, Jeremy.
Hi, Gethin.
This is a bit sort of off topic, the question, but at the Montreal meeting, Jonathan Glass showed that they got some post-mortem samples from patients who'd been receiving the failed C9orf 72 treatment, and even though they saw good penetrance into the CSF, when they actually looked in the brains into the neurons of the patients, they saw very poor penetrance of the treatments. So I'm just wondering if anyone has looked at that for the SOD1 treatment. I know tofersen works, so perhaps obviously there's a lot less post-mortem tissue available. But has anyone an idea of actually the reduction in SOD1 actually in neurons of treated patients rather than the CSF?
Not as far as I'm aware. Obviously, those results are relatively new. That clinical trial, I think, was published in 2022, maybe. So I don't believe anyone has looked at it, but that's not to say that it's not going on.
Thanks. Susan, you should be able to enable microphone and camera if you wish.
Thank you for your talk. I was just wondering if you've used any of your treatment approaches in other misfolded proteins such as TDP43, which should obviously be more relevant for the majority of patients.
Yeah, we think our lab is mostly focused on SOD1, particularly Justin was very focused on SOD1. So we've started there because I guess we know the most about how SOD1 misfolds and the toxic species. But yes, we are trying to develop approaches for TDP and fuzz or fused in sarcoma.
Good, thank you.
Thanks. I just have a question from the chat from Rebecca Zangil. She says, great talk, Jeremy. Some great data from the misfoldable transgenic mice reducing insoluble SOD1 in the brain. She asks, are you hoping to get target engagement with the brain plus spinal cord with an AAV approach?
Yeah, we are hoping to get a lot better spinal cord expression of the misfoldable so that it will be able to have better target engagement there with that pathogenic SOD1, which is primarily localized in the spinal cord of those SOD1G93A mice.
Thank you so much. Thanks, Jeremy. I think we might wrap it up there. So thank you so much for your presentation today. It was really interesting to hear how the lab is tackling SOD1 in MND from so many different angles to create a really multifaceted attack against its role in disease progression. And I think some similar principles and idea can really be applied from this approach to other MND relevant genes and pathways. So if there's anything else anyone would like to follow up with Jeremy, you're very welcome to get in touch with him directly or with us at FightMND if you would like us to help connect you. If you have any colleagues who weren't able to attend today's talk, it was recorded and you'll be able to view it on our website in a few days' time. So thank you so much for joining us today for the May FightMND Scientific Seminar, and we hope you've enjoyed today's presentation. The next monthly seminar will be on Tuesday, the 24th of June at 12 p.m. Australian Eastern Standard Time, and we'll release the details in the next couple of weeks, so keep an eye out in your inbox for this. Until then, all the best, and thank you, and we'll see you next time.